Hello everyone, and welcome to the webinar. My name is Sofia, and I'm a colleague to Casper Salinas. I work in communications, and I'm here to help out during the webinar. And Casper is sitting right here beside me, and he's ready to get started with his presentation. Casper, you will be presenting for approximately 20-25 minutes, and then we'll turn to questions. So if you have any, make sure to send them through to me via the chat function here in the webinar, in the meeting, and then I'll pass it over to Casper after his presentation. So that's it from me now. Over to you, Casper.
Thank you very much, Sophia. Yes, I am Casper. I am a biomedical graduate from DTU, Technical University of Denmark. After my education, I did a PhD, and that was mainly within image analysis of microscopy images of mouse brains, and that was in a preclinical drug development setting. Today, I work at Gubra. I started here in 2018, and I'm now a department manager of the AI technology department. So today, I will give this webinar, and I will start by introducing you to some of the 3D data sets that we analyze here. I will introduce you to the steps we take when we build and use AI models for this analysis. And I will do it by going through a case study related to a Parkinson's disease model. At the end, I'll provide a short summary. So yeah, thank you for tuning in, and let's get to it. So here on the first slide, I would like to explain a little bit about the imaging system that we use. We use light sheet fluorescent microscopy, and the illustration here shows a light sheet with the yellow line you see here. And when the microscopy session starts, the sample, in this case a heart, will be moved incrementally through the light sheet, and for each step we will take a 2D image. And you will see a short illustration here. And for each position, the camera perpendicular to the light sheet will acquire one image. I will now play a movie. You see here to the right on the screen, and this movie is of a mouse heart stained for collagen fibers. You see the images here, and as we zoom in a little bit, you can get an idea about the resolution we use. The resolution is normally in micrometer scales. It can be from a single micrometer up to maybe 20 micrometers. Here you see a maximum intensity projection, and you might see here in the middle some fibrosis built up in this mouse heart. On the next slide here, I want to give you an idea about the size of the datasets that we work with. To the left, you see a mouse brain. This mouse brain has been stained for neurofilament. It's a marker for the white matter tracts in the brain. This image was acquired in 5 micrometer resolution in the XY plane, and with 10 micrometer step size between the images. A one-channel recording of this would be around 20 gigabytes of data. To the right, you see another example of a mouse brain. This is stained with the TH antibody, showing a precursor for the neurotransmitter dopamine. This one we acquired at higher resolution, 1.4 micrometer in the scan plane, and with a 4 micrometer step size. A recording like this will take up around 300 gigabytes of space. You see, you also get a lot more details here. You can appreciate both some axons and some cell bodies in this image. So now I want to talk a little bit about the steps we take when we want to build an AI model to analyze datasets, as the examples I just showed. First, we will do an initial data exploration, and at this point we want to get the first feel for the data, and we want to see what types of signals we have to find. They can be circular, elongated, other things. We also want to get an idea about the sample-to-sample variation. Are there any artifacts we have to take care of, and so on. After this phase, we are ready to select the training data. This is the data that we will need to annotate to provide for examples to train the AI model later on. The third step is annotation of these training data. Here you can turn to fully manual annotations, but this is a very inefficient process, so we will give an example of a semi-automatic approach for this annotation. When this is completed, it's time to actually train the model, and I'll tell you about the framework that we use for this. Once the model has been trained, we have to validate it in the data that we train, of course, but also we'll talk a little bit about how we validated future studies if this model is still applicable. All right. So now to the case study where we give an example of the 5 steps, and this relates to a preclinical mouse model of Parkinson's disease that we work on here to establish. The model is an in vivo model where we inject some preformed fibrils into the brain, and these will form aggregates of alpha-synuclein, and this alpha-synuclein will spread throughout the brain over time. So the setup is that we inject the animals and wait a certain amount of time before taking out the brains. Those we stain with an antibody and later on image in the imaging system I just showed before. When we have done that, we will get an image like you see to the right now of the alpha-synuclein spread in the mouse brain. The goal of our segmentation and quantification will be to segment out this alpha-sense sequence signal and visualize and quantify it in various ways. All right, so when we start the first step, which is the initial data exploration, we look into the study groups because these will normally have different appearances of the signal, and we want to explore examples from all the study groups. In this case, the experiment was fairly simple. It had 2 groups with 4 animals in each. One group was injected with the preformed fibrils, and after 8 weeks, the animals were terminated and the brains extracted. In the other group, the alpha-synuclein could spread a little bit longer. It was spreading for 12 weeks before termination. Now, you could say you could start opening up image render and look at these files manually, but we prefer to take a computational approach to the further data exploration. And on this slide, I have a big purple box here to the left that represents the raw data volume. In the example of the alpha-synuclein brain, we imaged this in 2.8 times 8 micrometer resolution, and that gives an approximate voxel size as written on this slide. As you see, it's a big image volume. So the first step we do is to divide it into to some subvolumes. In this case, hundreds of subvolumes. These subvolumes are 224 by 224 voxels and 96 images in the stacks. This, again, we further reduce by taking out the middle image of each subvolume that you see here to the right. These images we call 2D patch previews, and these are the images that we will now do some computations with. We have built a vector embedding framework where we utilize some pre-trained AI models to help in our exploration. We use Clean Vision, which is a tool to find low-quality images in your dataset. Low-quality images in this case can be images that are very dark, it can be blurry images, images in general with low information. The other framework we use is a CLIP, a model trained by OpenAI, and that's a text image model that was built by taking 400 million images of the internet and pairing them with their text descriptions. And using the CLIP model to embed our images is a convenient way to go from an image space into a vector space. And when we're in this vector space, we can do some dimensionality reduction to explore further. This we color code in different ways, and I will show an example. And now we'll see here a movie play of one of my colleagues who used this app we have built to explore the data. Each point in this movie represents one image. First, you can choose how many images you want to have in the viewer at a given time. All the images is normally a bit too many, so here we use 10% of the 2D patch preview images. Now they are color coded by their group, so the blue, red, and green color. Now we turn to the Clean Vision embeddings, and here we could, for example, investigate how much information there is in each image. Here, a dark blue point is an image with low information. To the top left, you will see an image preview corresponding to each dot in this viewer. Now when we click on images with more information, like the yellow points here, you see to the top left that these images has more interesting information. To select what images we want to take further for annotation, we start thresholding on some of these parameters. First, we threshold on the information score. Here turning now to the blur score. In this example, there was not many blurry images, so we continue to the dark score and again filter away some images that are very dark. When this process is complete, you see we have reduced the number of potential images to annotate. Very nice. So now we got a lot further in getting ready for annotation. However, as you can still see, we still have a lot of images here, and this is, if you look to the top right, only 10% of the total 2D patch previews. So we want to reduce even further, and the way we do that is to use a k-means clustering approach. And we do this to make sure that we select images that represent the full distribution in this vector space. So in this drawing here, you see the big black dots, which are the k-means centroids computed. What we then do is we choose the closest point to each centroid. In this example, we chose to choose 20 images for further annotation. That can vary between studies, but 20 is a good starting point. So just referring these dots in the app back to the actual images they represent, the 2D images that were embedded again corresponded back to a subvolume, and it's these subvolumes that we now want to annotate. And again, in this case, we now need to annotate 20 subvolumes, each having the size of 224 by 224 times 96 images. This is in 3D will take a long time to do manually. So as I alluded to, we will present a method for semi-automatic annotation of these. One tool we are investigating is using Fiji together with a LabKit plugin. Again, I have a small movie here demonstrating how this looks. And you see here we have decided to annotate 3 classes. The red class that is being labeled now, this is the main signal class of the alpha singlet, and this is drawn here. We also have a tissue class. This is the green color, and this is to annotate the tissue that's not alpha-synuclein, but still tissue inside the mouse brain. The blue color that is not used in this video is for overall background outside of the mouse brain. The idea is now to draw some examples. You see here a couple has been drawn on this image, and then we can use the LabKit plugin to predict the annotations for the rest of the image. And that's a very efficient approach. In the beginning, the prediction will not be perfect, so you can provide more examples. And after a few iterations, normally we are happy with the annotations. In general, if we are to annotate a volume of these 96 images, then normally providing examples of only 5 of these images is enough, and then you could say the rest of the annotations for free using this plugin. So that was kind of the first 3 steps that we take: the initial data exploration, selection of the training images, and now also we have annotated the images. So now we are at the point where we're ready to actually train the AI model. And here you need some different things. You need some hardware. We use on-premise servers with a GPU installed. For the deep learning framework, we normally use Python in combination with TensorFlow. And from here on, there are many choices you can take when you want to build your AI model. Normally, we start out testing a 3D version of the UNet, and for loss functions, we try the dice laws in combination with the focal laws. And for the optimization algorithm, normally we start out with the EDA optimization algorithm. To get the best model, you might need to try different combinations, and to keep track of what you have tried out, we use MLflow to keep track of these things. I have now here on the next slide an example of a training curve for this alpha-synuclein model, what you see here is that in the beginning of the training, down here at epoch 0, the performance of the model is not very good. So we measure the average intersection over union score, and this is a measure that shows you how much of the annotation overlaps with the output of the model. And in the beginning, it's not a very good correspondence or overlap. As we start to train the model and go through the epochs, you see in the yellow curve here that, and also in the red one later on, that the performance improves. And in this particular training, after around 50 epochs, we have gotten to the maximum performance with this model. And here we have a score of around 0.8, which is a fairly good result. Also keeping in mind that the manual annotations or semi-automatic annotations are never perfect as well. So in this case, we chose to move forward with this model, and the next step is to do a manual evaluation on the patch images we selected from the embedding framework. And you see here with a purple overlay that the model performs quite well. So here we have overlaid the model output segmentation on top of the raw data. Very good. So at this stage, you could say now we have a model. We have tested it in our study, and it performs well. So we're very happy. One thing to keep in mind now is that if we want to use this model in the future for studies alike, it's very tempting just to take this model and apply it again. And in some cases this will work, but it's also important to realize that in other cases the model performance might not be as good as when you initially trained it. And that can be due to many reasons. For example, you could have a different study setup. So maybe the alpha-synuclein did not spread in the brain for 12 weeks, maybe it was 12 months, meaning that it could go to new places and have different appearances. Maybe you ran out of antibody, you had to buy a new batch, and that could give some variations in the appearance. So for these reasons, it's important to have a strategy for evaluating if your model still performs well in future studies. And to give an example of this, I brought an example here for the next study in this pursuit of a preclinical Parkinson's model. And in this case, we added 2 more groups. One group was, you could say, a negative control group here on the top, where we did not inject any preformed fibrils, so there should be no alpha-syn spreading. And we also had another group where we injected preformed fibrils, but took the mice down after 1 week compared to the 8 weeks and 12 weeks in the previous study. And to get an idea about if our model from before will perform in these animals, we return to the same embedding framework as before. In this time, I have color-coded the images from the 2 studies as batch 1 and batch 2. So the blue color are images from the initial 8 and 12 week experiment, and the red colors are from the 1 week experiment. And what you see here is that these points representing the images are very intermixed and lie around each other. So that's an indication that the model previously trained probably will also have a good performance in this new data set. So having this confidence, we applied the model for the full data set. And again, as before, we also did some manual inspections of the segmentation quality. And also here in the second study, we were happy with the performance. Again, the purple overlay here shows the model segmentation output. All right. So on this slide, we took the segmentation masks and we extracted some endpoints from the segmentations. So in this case, we on top of doing the alpha-synuclein segmentation, we also did an atlas registration, meaning that we inferred a brain atlas onto our samples. And by doing this, we can make some visual average representations of the alpha-synuclein spread. I provided here 2 examples. The top row shows coronal sections taken in the front of the brain around the primary motor area, colored here in a green overlay. And what you see here is that in the non-injected group, the alpha-synuclein levels are low. When we look in the 8-week groups with the preformed fibrils injected, we see a spread. We see it in both sides of the brain, and we mainly see it in the outer cortex area. After 12 weeks, the spread seems to be even larger. We also have an example from the middle part of the brain here, highlighting coronal sections around the substantia nigra. And again, you see here that in the non-injected group, the signals are the lowest. If you move to the 8-week group, you will notice again a large spread in the cortical areas, but also around the substantia nigra, which are located here with the dashed lines. What you may notice here is that the spread of alpha-synuclein is not symmetric. The spread is larger in one part of the brain, or one half of the brain, compared to the other. And this also makes sense since we did a unilateral injection, meaning that the preformed fibrils were injected in one side of the brain only. After 12 weeks again, we see a larger spread of alpha-synuclein. Another way to represent these findings are with bar plots, as shown here. So here we counted the number of objects with alpha-synuclein in different brain regions, and we normalized it by the area, the volume of these brain regions. So again, you see the same picture, that after 8 weeks, the spreading has increased compared to the non-injected group, and spread even more after 12 weeks. And this kind of wraps up the case study I brought for this webinar. To summarize the steps we take in training and using AI models, we rely on image embeddings from pre-trained models to do our initial data exploration. We use a clustering approach to select the images we want to train, and then we use a semi-automated approach for the training. We use Python and TensorFlow with the 3D unit to do the initial training of our model. And when we want to validate, we again use this embedding app that we built. In this case, the model that was trained in study 1 also worked in study 2. But if you are in the case that the model does not perform well in a future study, you can see here, I put in a kind of dashed arrow here, representing that what you then do is that you kind of start the circle up once more. And from your new study where your model did not perform, you again select some new training images and annotate those, retrain a model, and then it will usually work quite well. So we have now been using this setup to train our models for a couple of months. And we've been using it for the case I showed here, but also for other cases like blood vessel segmentation and analysis. We've done that in muscle tissue and also in brain tissue. And for my last slide today, I'd like to show you one of my recent favorite segmentation masks, which is exactly this, blood vessel segmentation in the mouse brain. And you can really appreciate the complexity of the vascular network in the brain. And with this last movie, I end this webinar. And as Sophia mentioned at the beginning, I will be very happy to take any questions you may have. Thank you.